Okay, great. Sandra, it's great to hear that you made it. So I'm Pat, for those of you who don't know me, I'm Patrick Grohar, and I'm at the Children's Hospital of Philadelphia. And this is really a fantastic lineup to start this meeting. So we will start with introductions in a minute, but Sandra, do you want to introduce yourself as well?
I do. Thank you very much, and thank you for the invitation to chair the session. I again would just like to congratulate Ita and everyone from Spain for organising such a wonderful meeting with such a great diverse array of talks. So I'm Sandra Strauss, medical oncologist based in London, Ewing's enthusiast, and I lead the Ewing's consortium, and it's wonderful to chair the session with Patrick and with such esteemed speakers too.
Great. Yes. Again, thank you to the organizers for what a wonderful meeting. I'm really excited about it. But I think we lead it off appropriately with Olivier. So if you don't know who Olivier Delattre is, you're probably on the wrong Zoom call. So you should probably sign off right now. But first, let me just give a brief introduction. Many of you know Olivier's scientific contributions to the field. A couple of things that stand out for me are just his ability to publish consistently in the top tier journals throughout the years. But also, I think two other things that stand out are one, he leads two other fields too. Like it's not just Ewing's, he's also a leader in neuroblastoma as well as rhabdoid tumor. And to me, this has always been particularly impressive. And lastly, I think we have a really nice community in Ewing Sarcoma, and I think it's based on the leadership of the group, and Olivier and Heinrich and Lee Hellman and Paul and many of the people who've been doing this for years have really made an inclusive community. So I think that it's a really wonderful thing. So Olivier, I'm going to stop rambling and turn it over to you. Thank you. Oh, one other thing, for those people with questions, if we have time at the end, please just type them in the chat, because I don't know if we got very many questions in the first couple sessions, and we'll field them as moderators.
Okay, can you see my slide? And can you hear my voice?
Both. Yes. Check and check.
Yes, so that's great. Thank you very much, Patrick, for these very kind words. And thank you very much, Uta and colleagues, for organizing this fantastic symposium. And it's a real pleasure to see so many good friends on the screen, unfortunately. And hopefully, in the next few months, we'll be able to meet in person. So I think, Uta, you asked me to give some background on the history of EWS-Fly, and I will do that, as well as maybe also talking a bit about new data that may be of interest for the community. So the story of EWS-Fly begins close to 50 years ago with two cytogeneticians, Alain Orias and Claude Turcarel, who described this translocation between chromosome 11 and chromosome 22 in Ewing sarcoma. And I think this was among the first recurrent genetic abnormalities to be observed in solid tumor. It was quite known in leukemias or lymphomas, but not in other kind of solid tumors. So my job at this time as a PhD student was to try to understand what was going on with this translocation and what was happening at the level of the translocation breakpoint. And at this time, we didn't know a lot about the human genome. We only had a very few probes on the chromosomes, maybe 15 or 20 on chromosome 22. And the job was to do some what we call chromosome walking, so starting from probes on chromosome 22 and trying to find out the breakpoint. I will not go into the details of this endeavor, which was quite long and time consuming. But ultimately, at the end, we could identify the breakpoints and show that as compared to germline DNA, tumor DNA was rearranged using this old technology, Thousand Blood, that I guess many new young investigators have not even heard about. And it took us about 5 years with 5 full-time persons to achieve this goal, to clone this breakpoint. And it's always interesting to see that nowadays you have the same result in only a few hours with this kind of device like a NovaSeq. So in Ewing sarcoma, the most frequent translocation is EWS-Fly. The position of the breakpoint may be a bit variable from one tumor to the other and lead to variable exon composition of the fusion transcript and of the protein. But what is important is always the 7 first exons of EWS are included, and the last 2 exons of FLY1 are included in the fusion. And there does not seem to be any prognostic significance of this variability. And in addition to FLY1, some other translocation have been also described with ERG, which is very close member of the ETS family, very homologous to FLY1, which is involved in around 10% of the cases, as well as other rare fusion with other members of the ETS family. Altogether, this genetic abnormalities with the slight variation I indicated is really pathognomonic for Ewing sarcoma. And it does not seem that this variability have any clinical consequences. In terms of what happens, what it leads to this fusion protein. So the EWS gene, which is also called now EWS-R1, in our initial description, in fact, EWS-R1 was only the position of the breakpoint within the EWS gene, but nowadays the gene is called EWS-R1. Anyway, it has, we don't know a lot yet about what is the function of the normal wild-type EWS-R1 protein. What is known is that the CTER is an RNA binding domain, able to bind RNA through the RNA recognition motif. And the N-terminus of EWS is what is called alternatively a prion-like domain, an intrinsically disordered domain, or a low complexity domain. And I will come to this in a minute. FLY1 is a transcription factor of the ETS family with a DNA binding domain, DBD, at the CTER, and a transcription regulatory domain at the N-terminus. And as a result of the translocation, there is a fusion gene coding for a chimeric protein, which has the DNA binding domain of FLY1 and this prion-like domain of EWS. And such prion-like domains have are the object of considerable interest presently, because this domain, and this is something which was quite well known by the physician, but only get to the biological field quite recently, is that such domain that are represented by this pink domain in here, this kind of domain has the ability to form aggregates when the concentration increases. So it's a concentration dependent aggregation. And this aggregation can be either through homotypic interaction, so with similar domains, or also with heterotypic interactions. And such droplets are alternatively called condensate, aggregates, granule, or membraneless organelles. And they seem to play a considerable role in the biology of the cell, being able to create some organelles that is strictly dependent upon the concentration. And why is this important for EWS FLY1? Is that the way we think EWS FLY1 is acting now is that it is able to bind to this GGAA repeat shown in this slide. And such repeats are usually in the completely closed chromatin conformation in normal cells. And upon EWS, the translocation and EWS FLY expression, EWS FLY can bind to this GGAA. And what is thought presently is that this binding leads to increased concentration of EWS FLY at the level of this microsatellite that can thus lead to homotypic interaction with the wild type EWS, as well as heterotypic interaction with many other member of the transcription machinery, chromatin remodeling machineries, histone modifying enzyme, and so on. And as a result of this, such microsatellite sequences are transformed in true and answer sequences that are able to activate genes in the neighborhood. And this is something which is very important for Ewing sarcoma biology, because this is somehow an epigenetic disease where EWS5 binding to the thousands of microsatellite sequence throughout the genome can completely reprogram the cells. So EWS5 through this binding, either to microsatellite sequence or ETS sequences, can alter a number of processes in the cells. And obviously, across the past years, many groups worldwide have searched for downstream pathway activated by EWS5 that could be targeted by small molecules or antibodies or whatsoever. And I'm shown here is the Igf-1 pathway for which Katja and Patrick, and as well as my lab, has done quite a bit of job. And the Igf-1 pathway is obviously a very interesting pathway to target in Ewing cells, even though it seems that the cell can rapidly escape to Igf-1 blockade. Other pathway, I will not mention all of them because they will be discussed throughout the session, and I'm sure that Heinrich will develop the network of EWS5 activity, and Thomas also will speak about this. But the cyclin D1 and the cell cycle related proteins that are activated by EWS5 are important, and there are some ongoing trials with CDK4 and CDK6 inhibitors. One thing that is important to notice for Ewing sarcoma is that it is not part of any predisposition syndrome, at least to my knowledge. Ewing sarcoma do not occur in predisposition syndrome. Yet, it has been described for quite a long time. And the first one to describe this was Joe Fromeni from the Lee and Fromeni syndrome, that Ewing sarcoma is much more frequent in population of European descent than in population from African descent. And this was nicely investigated by Logan and Josh in a recent paper. And if you see on the right, this is very striking that when you look at the USA, in the USA, the African American population, the white population, the ratio of Ewing sarcoma is approximately 1:8. So clearly showing that even though Ewing is not in predisposition syndrome, there is somehow a susceptibility for Ewing sarcoma, which is highly population dependent. I will not go into the details, but this was investigated by my lab in collaboration with Stephen Shannock's lab and across the past year. And it's very clear in doing a GWAS, so comparing the genome of Ewing sarcoma patient and non-Ewing matched population, that there are at least 6 loci in the genome that are strongly associated with Ewing sarcoma predisposition. And susceptibility, because this is quite a weak predisposition, but this is a susceptibility. And investigating those different loci, we think, are highly important to understand the biology of Ewing sarcoma. Just to come to our current working model for Ewing sarcoma, is that the cell of origin of Ewing sarcoma, I think most people will agree that it's some kind of mesenchymal stem cell that can be transformed by EWS5. But this transformation is dependent upon polymorphism in the mesenchymal stem cell genome that favor the activity of EWS5. And so, second aspect, which is important, is that Ewing cells can oscillate between two states with a gradient between cells that have high EWS5 activity and that are highly proliferating, and cells that have a lower EWS5 activity and which somehow recover their property of the mesenchymal lineage and have the ability to migrate and to to migrate. And this fluctuation of EWS5 activity is probably dependent on a number of different factors. And Heinrich, I showed this with hypoxia, and other have showed that antagonisms like Wnt and EPO can counteract EWS5 and may lead to some EWS5 low phenotype. And we believe that this plasticity between the two states is very important. And STAC2, which is one of the most frequent secondary alteration in Ewing sarcoma with loss of function, and this was shown by a paper by Didier in my lab and the group of Kim Stegmaier. Somehow the deletion of STAC2 pushes a bit the cells toward EWS5 low phenotype, which may account for the increased metastatic potential of those cells and their resistance to treatment. In terms of one thing which I think is very important and for which the working together of the whole Ewing community is very important. And I know that you have been leading a number of studies on this, and this is very important, is how could we best stratify the patients, the Ewing patients for therapy? And for this, we will hear a bit about this in this meeting. Secondary alterations are important, and at least 4 of them have been associated with adverse prognostic: the unbalanced 116 translocation, the CDKN2A deletion, STAC2 mutation, and P53 mutation. And I think it's very important to see how much these alterations combine to confer adverse prognosis in Ewing sarcoma. And second aspect that we will hear a lot about also is circulating tumor cells or tumor DNA, and which also is a biomarker that may be extremely useful for Ewing patients. And we will hear from Marcus Metzler on this, and those biomarkers are certainly very important. Now I would like to turn rapidly in the few last minutes in a new finding in my lab. So I told you that EWS5 is able to find these microsatellite sequences. And this is shown on the top here where you have a GGA repeat, and ChIP-seq clearly show that EWS5 can bind to this. And when EWS5 is inhibited by treatment with DOX to induce a small hairpin RNA, this signal completely disappears. And some of the binding of EWS5 on those repeats are in completely desert region of the genome. So they do not activate any known genes. But what we showed is that when EWS5 can bind, it can attract the histone-modifying machinery, as shown with the K27 acetylated mark, methyl-3 mark, and can thus induce a completely new gene, which is completely unknown in any database, and which is shown here with different exons. So with two students in the lab, Julien and Olivier, looked at this and clearly showed that those genes are really highly specific for Ewing sarcoma. They are not expressed in other cancer in all the TCGA database, apart for a few outliers, and they are not expressed either in any of the normal tissues. So this is also shown here where you have the Ewing sarcoma as a red spot in the middle, which is the expression of different neogenes that we showed. And you see that in other cancer or in other tissue, you only have very weak and sporadic signals. So showing that these genes are extremely specific for Ewing sarcoma. With the model for the expression showing here is that in non-Ewing cells, you have those microsatellites that are completely in the chromatin closed conformation. And upon EWS5 in Ewing expression in Ewing cells, EWS5 can either activate known genes with these enhancer mechanisms I told you about, but can also induce the expression of completely new genes. And what Florian and Céline investigated in the lab is to ask the question, are there some of those neogenes potentially translated into protein? And to make a long story short, they did ribose sequencing as well as a lot of mass spec analysis and could detect, as has been shown for other linked RNA, that indeed some expression of peptides can be observed in this for this neotranscript. So meaning that these neopeptides are absolutely exquisitely specific for Ewing sarcoma. And we believe that this is important because our question now is, might these peptides encoded by the Ewing-specific neogenes be instrumental for immunotherapy approaches, either by vaccination or by cell therapies? And this is really something we think may be very important for the field. And here I will stop, thanking all my colleagues, thanking the three guys on the top, three former postdocs that have now their own group in Zurich, in Lyon, and in Heidelberg, and also showing Julien and Olivier, who have done most of the job for these neogenes in collaboration with Josh Waterfall's group. And I think I'm finished now. Thank you very much for your attention.
Thank you, Olivier. As usual, a wonderful presentation with some really exciting new findings. I can't wait to see that manuscript. In the interest of time, I think we're going to keep moving and invite the next speaker, Tomas Greenwald, to present. So Tomas is a pathologist and the division head at the Haupt Children's Hospital in Heidelberg. He runs a broadly focused research group and has really published prolifically lately. So Tomas, on that note, please take over.
So, can you hear me? Okay.
Perfect.
Yeah, thank you. Thank you very much for the introduction and the kind words. I also see me a bit in the genealogy of Olivier and would like to thank him also for his support in the past years and the very good collaboration that we kept from ending my postdoc. Now, I started my postdoc almost 10 years ago in his lab. So I was asked by Uta to give a talk about the best of biomarkers and targets in Ewing sarcoma, and I have 15 minutes to wrap up 50 years of research. And for that reason, I have to do a disclaimer. So it's a bit impossible to cover the entire field in 15 minutes. And all I present now and the selection I had to make, unfortunately, is purely subjective. And I apologize to all those who may not be covered here now with their work. It's not a sign of disrespect, just a matter of time. I could not capture everything, but I would like to catch up with something that we have heard today, and I would like to give a bit of historic perspective. I'm not a dinosaur yet, but I at least try to give some sort of this respect to this perspective. So more than 30 years ago, IGF and IGF signaling was discovered as a potential major driver of Ewing sarcoma. And this group in US showed that blocking this pathway has negative impact on the growth of the cells. And this was closely followed then by developments in Europe, mainly driven by, by the Italian group at the Rizzoli around Katja Scotlandi and Nicola Baldini, who showed that indeed IGF signaling might be a valuable target in also in vivo, and that this can be blocked by monoclonal antibodies and has growth restrictive effects. And after that, we have seen that there was a lot of biological studies ongoing to underpin the mechanisms, and it has been shown that IGF-1 and also IGF-BP3 are somehow related, but counteracting regulators of IGF-1 signaling activity and may be used even as predictive biomarkers for a therapy. This goes on then with a burst of new papers coming out in the next decade by Olivier's group, showing that EWS-Fly suppresses IGF-BP3 and therefore enhances this IGF signaling. Other groups have, it's a bit slow, then developed inhibitors, tested inhibitors preclinically in different models, different approaches. And this all culminated, and I think for me, these were the two major studies at that time, and the two back-to-back published JCO papers showing that there is activity even in refractory patients. And I think it was the study published here by Heribert Jürgens that showed that IGF-1 may have predictive value We have learned today that in metastatic disease, a monoclonal antibody in maybe not pre-selected patients may not be successful. But I think this is not the end of the IGF-1 story. For the sake of time, I have to jump to the next topic, and I can only touch them superficially. But one thing I want to mention today briefly is a thing that also struck me when I was a PhD student in Stefan Burdach's lab is the story about the physical interaction of EWS-FLI1 with RNA helicase A. We all know that this facilitates the transcription of specific target genes, that this can lead them to the development of a specific inhibitor. And I think we will learn more about this today by subsequent speakers, Jeff Turetsky, Stefan Zöllner, and Joseph Ludwig. So I will not talk about this in detail, but I just would like to mention that the Nature Medicine article shown here was my first Ewing sarcoma paper I presented in a journal club. And this was, I was looking for a good paper, and this just came out, and I still love this paper. And I can just recommend all young people to read this, as I think it's a very good story. And who followed this for the last 10 years, how the team around Aykut and Jeff Turetsky developed further this inhibitor and pushed it to the clinic. This is really an amazing story how this should be done. I have to jump again in time and space. Here we come. I think this was the first description of a recurrent mutation next to EWS-FLI. STAC2 was reported here to be present, mutated in 21% of the tumors, as you can see here in the IHC stains. This leads to a truncating mutation and loss of expression and therefore also function. This study published in Science was 3 years later, followed by, I could say, 3 back-to-back papers, or 3 papers that came out almost at the same time from 3 different groups. We know all this, we know all these studies that first of all confirmed this finding, found also that other mutations are not very frequent, and that really Ewing sarcoma is about EWS-FLI and possibly STAC2. And as Olivier has mentioned, another back-to-back story a couple of years later, the group of Kim Stegmaier and Brian Crompton, as well as of Olivier with my friend Didier here as the first author in this cancer cell paper, shed light on the underlying mechanisms, which is very complex, so I can only briefly touch upon this. But as Olivier said, this pushes the cells into a more mesenchymal and therefore more metastatic and aggressive phenotype. Up. So, sorry, my computer is kind of stuck all the time. Yeah, but this is not the end of the story. So I think there is also potential for STAC2 not only being a prognostic biomarker, but there is also maybe room for interfering with STAC2 activity, or in terms of synthetic lethality also, because it confers synthetic lethality to a specific inhibition of STAC1. If this will ever happen in Ewing sarcoma or be feasible is another story, but I think this is very interesting that we do not only have here a potential prognostic marker that is mechanistically very well explored at the moment, but also an indicator of that it can lead to a synthetic lethal phenotype. Yeah, my computer is stuck. Yeah. This paper, you know it all, it published Nature 2012. I just started my postdoc in Olivier's lab, full of energy, wanting to cure Ewing sarcoma. Then this paper came out showing that imatinib works against BCR-ABL, who could ever tell, and surprisingly, that olaparib works in Ewing sarcoma cells. And I said, okay, I should have taken another career on another topic, because Ewing sarcoma is maybe already cured before I started my postdoc, so there's nothing left for me to do. And this was then more or less confirmed, or came out at the same time by a prostate cancer group here in Ewing sarcoma, the same thing, a bit more in-depth focus on Ewing sarcoma. And this, yeah, I still kept on doing Ewing sarcoma research, and it not took so long time to come, unfortunately, to the conclusion, sorry, my computer is stuck, that yes, this was sadly for the patients, maybe good for promoting my career, but I would have preferred the other way around, that olaparib was not the breakthrough we were perhaps hoping for. At least as a monotherapy, it was not really effective. And please, clinicians, correct me if I'm wrong or if interpreting it wrong, but I think this was a bit surprised that we had such a strong in vitro finding, and then in patients it turned out to be so little effective. At least I didn't really expect that. But I think many of you know that there is now stories ongoing and studies ongoing that test PARP inhibition with traditional chemotherapeutics. And there are first studies coming out showing that this approach is safe and that there is maybe also some signals for activity and the different ways of combining now the drugs. And I think also, like with the Igf-1 story, this is not the end of the story. I think we have to better understand when, how, and who should be treated with olaparib in which combination, and that there is really some potential for maybe a certain subgroup of patients that may benefit from this. You all know maybe this paper. It shows that PARP inhibition can also be used in combination not only with classical chemotherapeutics, but also with a more targeted approach, and maybe at some point the combination of olaparib or PARP inhibition with classical chemotherapeutics and also targeted inhibition, for example, here of CDK12, may pave the way for better treatment protocols. There are some, I would like to use the last minutes for some potential rising stars. I separated this in prognostic, predictive, and liquid biopsy biomarkers and potential targets. Rising star for studies that are almost 10 years old, but just shows how much it may take. You know the story of Enrique de Alava's lab, that not only point mutations, fusions may have maybe prognostic role, but that it is also here gains in copy number losses. Here in this particular case, 1q gain. There's another paper coming out maybe almost at the same time. Sorry, my computer is stuck again. Yeah, here about this story, I think Katja Skotlandi will talk about this later. But also microRNAs seem to have a prognostic role and also a mechanistical role in chemosensitivity and promoting progression of Ewing sarcoma. And then part of my PhD project was at the same time to investigate the role of STEAP1. And we have shown that surprisingly high levels of STEAP1 are of good prognostic impact in Ewing sarcoma. And all of these biomarkers and some more are currently under investigation in the so-called PROVABIS project, which is prospective validation of biomarkers in Ewing sarcoma for personalized translation in medicine. And this is the consortium. And as I indicated, there will be more on this by Katja Skotlandi, probably at least on microRNA 34A. And I'm very curious to see what comes out in this validation. And we will hopefully learn a bit about this in the proximate future. One thing that came out from our group 2 years ago, and this was a bit surprising finding, you know that there were functional reports on SOX2, that SOX2 may be a common feature of Ewing sarcoma pathogenesis, and that SOX2 is expressed and confers an aggressive phenotype and some stem cell features. When we looked by SOX2 antibody stains and TMAs, we have seen that basically most of the Ewing sarcomas show no expression of SOX2 at all. And there was just a minority of patients that showed SOX2 expression, but it was not like in this extreme example. The few patients were just some nuclei positive. But however, when we looked here in 2 independent cohorts at the mRNA or at the protein level, once we had high mRNA levels, which was just in a minority of cases, or detectable SOX2 expression by immunohistochemistry, these were patients with a very poor outcome. When we further analyzed this together with Uta Dirksen, we found that this was among localized patients, so patients without any apparent other clinical risk factor. There was a subset, about 1/3 of those patients with very high levels of SOX2, which tended to have early relapses. And I think this could be a very easy to test biomarker, and hopefully in prospective settings, this can be validated, but this we need to wait for. Another study, I cannot go into details because I think Sue Birchall will talk about this later, is a very interesting finding, which is similar to the SOX2 finding, is the levels of neurexin. When you look at this paper, there is really dramatic differences in Kaplan-Meier plots. So I think this is also worth being prospectively evaluated. And again, here, it seems, if I've understood the paper correctly, that the especially for localized patients, this could offer an additional value to detect maybe a hidden high risk group among so-called low-risk patients that should perhaps get a different treatment upfront if we could identify them in the diagnostic biopsy. Now some potential rising stars, predictive biomarkers. This is also of my group. We've recently shown that there's EWS-Fly activates the expression of PRC1, which is not the polycomb repressor complex 1, but the protein regulator of cytokinesis, and that this ensures the correct separation of chromosomes during cell division. And when you interfere with this through blocking the upstream activating kinase of PRC1, which is PLK1, then this may interrupt this axis and then lead to aneuploidy and cell death. And we could show in vivo that when we treat mice with high that bear tumors with high PRC1 expression, that then we can, in a fraction of those cells, tumors achieve complete tumor regression. However, when we knock down the PRC1 associated GGA microsatellite and thereby reduce PRC1 expression levels, then we cannot cure these mice anymore. We see a little bit of a delayed tumor growth, but there was no tumor regression achievable. And we are currently trying to also evaluate this in more sophisticated models, PDX models, and hope that we can validate it and then try to see whether this could potentially serve as a predictive marker for PLK1 inhibitors such as volasertib. Just very briefly, for the sake of time, liquid biopsies, as Olivier mentioned, play a very potent role and promising role to be really very good for disease monitoring. And there are several groups, like the group of Brian Crompton and also Markus Metzler and Eleni Tumasu, who did really great contributions to this field. I will not talk about this in detail because they are going to do so later. And with this, I may close and again apologize to all people who I could not mention today, whose work was maybe just omitted. Blame it on my ignorance, and for the sake of time, I would like to acknowledge my team who helped me build up my new lab in Heidelberg. I can tell you there's more funny things to do than starting a new lab in the pandemics. But they were really a great help for me. And I also thank you for your attention and my sponsors for their valuable support.
Tomas, wonderful job covering all that ground in 15 minutes. Really great, as always. So thank you for that great presentation. We'll move right on to the next speakers, as Tomas alluded to, speaking about the potential liquid biopsy. So Markus Metzler is at the Friedrich Alexander University in Bavaria, and Eleni Tumasu is at the St. Anna Children's Cancer Center in Vienna. Markus, the first time I heard of liquid biopsy was at a Nuremberg conference in 2013 that someone was talking about earlier. And Eleni has has, I think, published a seminal paper on epigenetics in Ewing. So anyway, here, I'll turn it over. We can see the slides, but we can't hear you.
And now you can hear me, I suppose.
Yeah, great.
Thank you, Patrick. And now you hear the follow-up of this first impression on liquid biopsy and Eleni. We not only share this talk today, this is because we share the enthusiasm on liquid biopsy in Ewing sarcoma. We share many samples together and analyze them from different ways. And now I share the screen with you to show what get out of this. So we today talk about potential biomarkers in blood samples of Ewing sarcoma. And for the sake of time, I just tell you that we focus on cell-free DNA for a number of reasons. One is that the cell-free DNA is relatively stable in serum. It's comparatively high copy numbers in the serum present. It's released by apoptotic and necrotic cells, so somehow tells you about the response to therapy, and then just one translocation positive fragment per cell, so some kind of absolute numbers to count for. Why biomarkers in Ewing sarcoma? Obviously, there are no other specific serum markers available until now, but the characteristic translocation, as worked out before, is virtually present in all cases. So we thought it would be worth to combine these two situations. We need a plasma marker. We do have the translocation within the tumor cells, and they are genomically very stable, clonally stable, homogeneous within the tumor and over time. There are a few recurrent secondary mutations which could be used for assays, and we know that DNA markers are highly sensitive and dynamic because we use them in leukemia MRD diagnostics for quite a while. So the thinking was that circulating tumor DNA may serve as a molecular marker in Ewing sarcoma as well. And this is just one clinical example of a patient with a pelvic tumor under treatment with reduced volume and then a pulmonary metastasis, which was then again treated and reoccurred later. So in imaging, you have always the difficulty to exactly tell what this signal does mean. Is this a still viable tumor? Is this remaining tissue, or what happens in between? And therefore you see that the imaging does give you some information, but it would be nice to see what actually happens in between. And sorry, second moment where I don't want to answer the phone. That the circulating tumor DNA gives a relatively strong signal, although the tumor volume is rather low at the same time point. And therefore, we ask ourselves whether we just cannot use them to sequentially follow patients, but to have an impression on prognostic and predictive value of circulating tumor DNA. And we studied this in a group of 120 patients, which have been treated within the Ewing 2008 trial, and took blood samples in the beginning, before and after the first video block, and then before the second, third, and the sixth block, and after local treatment. These samples have been analyzed for the EWSR fly fusion gene, so on the DNA sequence, and this has been either identified from tumor tissue or later from the plasma fraction itself already, and then quantified by digital droplet PCR to give us a highly specific and high sensitive measurement. And this follow-up measurement has then been analyzed together with the typical clinical and molecular data in terms of the correlation between ctDNA level and the tumor volume, the known risk factors, and obviously also the outcome parameters. So what we did see is that there is a strong correlation between the fusion gene copy number and the tumor volume. The black dots are relapsed patients. They tend to have higher copy numbers. And there are just a very few cases with negative signals. These are the few ones which have been surgically resected ahead of the treatment. So this seems to be real. If we then separated the two groups from each other, the ones which did develop an event, a relapse of the tumor or progression later in the course of the treatment, then one could see that from the very beginning, they had different copy numbers, so tend to have a higher copy number, the ones with a poorer prognosis. already ahead of the first treatment cycle. Same holds true after the first treatment cycle, although it somehow already differentiates a little bit better between the both of them. And then only the ones which relapsed later, there are few cases which remained positive during the entire induction therapy. Interestingly, the correlation between this copy number of the fusion gene and the like typical risk factors like localization on the pelvis, large volume, metastatic, and osseous disease, they correlated in the right way how we expected it. Only in the respect of good response versus poor response, the good responders tended to have a higher copy number. And this was in the first thought somehow contradictory, because it thought that the poor responders are the bad guys, so they have to behave like the other adverse risk factors. But biologically, it's far more logical this way, because they they are degraded by therapy, and therefore they release more DNA into the bloodstream as the others one which are less sensitive. This could be also shown for the risk arm. So with increasing adverse risk, the copy number increases, and this is independent of the tumor volume and the size and the localization. So it's not just representative of the tumor volume, but this have kind of intrinsic information. And eventually, if we plot the survival, overall survival and event-free survival against the cases with low, medium, and high ctDNA levels, one could see that it splits up between the three groups. And in particular, in the patients with metastatic disease, there's a small group with very low levels which seem to have not that adverse outcome as expected for metastatic Ewing sarcoma, and the ctDNA copy numbers distinguish better between these three groups than most of the other typical risk factors. And the most informative time point under this treatment is the status after the second video block. So this is the strongest distinction between the good and the poor responders. So at this point, we summarize that ctDNA has a predictive and prognostic value in the context of the Ewing 2008 therapy. There's a high correlation with clinical risk factors and metabolically active tumor volume. The ctDNA levels provide an integrated information from multiple tumor characteristics. And we obviously following these mechanisms of release and correlation in preclinical models now in more detail. And we obviously plan and already implemented this in the upcoming protocols to measure also during the entire therapy and follow-up phase. And obviously we think it's a dynamic and non-invasive biomarker, in particular for monitoring relapse cases. But obviously this is not, this is the work of many people in my lab and a result of the very productive collaboration with Eleni and Uta. And so Eleni will show how our complementary approaches come to a common result. And since we focused on genetic features, Eleni will show that the epigenetic signature of Ewing sarcoma is also a very useful marker. Eleni.
Thanks, Mark. So you have to change the slides for me. Yes, thanks a lot. So similarly to Patrick, I also got interested in liquid biopsies by listening to presentations by Markus Metzler at the Ewing meetings. And back then, I never thought that I was never planning to go into the liquid biopsy field. But in 2017, we published a paper where we nicely described that Ewing sarcoma tumors have a very unique epigenetic signature. So it's so unique that if we didn't have the fusion oncogene to diagnose the tumor, we could have done it by looking at the Ewing sarcoma specific enhancer elements. So Olivier gave a very nice overview about this field. And we have also shown that this epigenetic signature can be linked to clinical data. So this was quite exciting. And at the same time, back in 2017, 16, there was some great papers in the field of liquid biopsy. So one was published in Cell by Jason Dewar, showing that by doing whole genome sequencing of cell-free DNA, you can infer nucleosome positioning genome-wide. So when I saw this paper that had nothing to do with Ewing sarcoma, knowing that we have a Ewing specific unique epigenetic signature, I thought, shall we try it? And I remember I was talking to Didier in one of the Ewing meetings, and he was nice enough to say, okay, I can give you a few of our samples and you can test your idea. And actually, Didier gave us our very first liquid biopsy sample back in 2018. And this was a sample from a patient that had very high tumor content in the circulation. And we were calling it the champion sample, because it was really fantastic with a lot of ctDNA. And we could actually confirm or prove that we could monitor or we could detect the Ewing sarcoma epigenetic signature in liquid biopsies. So based on this result, I went then to Markus and said, cool, it works. Can we share or can we get access to your precious sample? So then we started using a material that Markus and Uda have been using for the EFACT study. And in parallel, we were doing our assay on looking for the epigenetic signature by doing whole genome sequencing. And in the meantime, we got more samples from France, from Didier and Olivier. And also we invited also a group from Norway by Ketil Boyet that also contributed to samples. So I will not go into details in our, Markus, please, you can change the slide. So I will not go into the details about the study because it has been recently published in, next slide, please. Yes, it has been recently published in Nature Communication. So in just quickly what we have done, we have collected cell-free DNA from over 100 patients with Ewing sarcoma. We perform whole genome sequencing and we develop bioinformatic tools that only take into account the cell-free DNA fragmentation pattern and the Ewing sarcoma epigenetic signature. And what I can quickly say is that our assay, we can very accurately detect tumor. We can classify the tumors, so we can separate Ewings from non-Ewings. We can study tumor evolution, so we can see how the epigenetic state of tumors change over time. We can quantify tumor, and we can also monitor tissue damage due to therapy toxicity. So the assay works fine, it's great, we're very happy with it. Now the big question is this assay feasible in a clinical setting? So if you ask me, I would say yes, because this assay requires less than 10 nanograms of cell-free DNA. It profits from the falling cost for next generation sequencing. So now it costs a few hundred euros, like less than 300 euros to do whole genome sequencing at about 15x coverage, and most importantly, it does not require access of primary tumor tissue. So we do not have to know what's the break point of the fusion. So now the good question, and now this is our goal together with Uta, Markus, who is clinicians, and also with Christoph Bock, who is a great bioinformatician and he's expert in machine learning. Our aim now is to bring this assay into the clinic. You will all agree with me that implementing a new assay into clinical practice, it's a long road with multiple steps. There is a very nice recent review published in Nature Reviews Clinical Oncology that nicely summarizes what we need to do to implement liquid biopsies into the clinic. We are still at the very beginning. So we are at the very beginning of implementing our assay in clinical practice. So what we have to do now, so we have shown our analytical validity of the assay, but as a next step, and this is something we work now, is to establish this clinical validation. So to do clinical validation to prove its clinical utility. And as Uta mentioned earlier, we are very happy that we get a very generous grant from the Vienna Science and Technology Fund, so we can go on and do this now. Next slide, please, Marcus. So in ideal setting, as a biologist, not as a clinician, as a naive biologist, I just have here a figure that in an ideal work shows the time points during therapy of a Ewing sarcoma patients where I would have liked to collect blood and liquid biopsy samples. So, so assuming that we have this very nice cohort, we would like to validate, clinically validate our assay for 3 different modes. So we want to assess our assay as a prognostic biomarker at diagnosis. Then we want to validate it as a predictive biomarker during induction chemotherapy. And also we want to validate our assay as a molecular disease monitoring assay during maintenance therapy, detecting relapse early on. Next slide, please. So very quickly, just we don't have time. So we want to validate our assay as a prognostic biomarker in diagnosis. So we want to know how well does our liquid biopsy assay stratify patients in localized Ewing sarcoma patients into high risk and low risk groups. And also we want to see for those localized patients that relapse or metastasize later on, if we could detect occult metastasis based on tissue-specific signatures. Next slide. We want also to use our assay as a predictive biomarker for real-time monitoring of response to induction chemotherapy. So the question is, can we predict patient outcome based on treatment response dynamics? And what are the suitable time windows for starting adaptive induction therapy? And as a last point, in mode 3, next slide, please. We want to validate our assay as a molecular disease for molecular disease monitoring and early detection of relapse. And we want to see how does this compare, our assay compares to other conventional approaches. Last slide. So this would be, currently we're in the process of putting together our cohort. So we would like to collect material from at least 200 Ewing sarcoma patients. And we are very careful at this point looking also at their clinical history so that we cover as many possible clinical scenarios. So we want to have at least 100 patients that had metastatic disease at diagnosis and 100 with localized disease. And then we want to have also patients that relapse and did not relapse later on. One important exercise now we have to do is to collect also proper controls. So currently we are collecting material from healthy individuals. So ideally we would like also to match them in the age. So far we were analyzing cell-free DNA from healthy adults. It would be great also to go towards earlier ages. We also want to include in our analysis patients with other sarcomas, especially when we validate our assay for tumor classification. And most importantly, we would also like to have as controls patients that do not have cancer, but perhaps they have other conditions like inflammatory condition, because we want to make sure that our assay does not just discriminate healthy from not healthy, but our signature, our signal really discriminates cancer from the rest. And last slide, please. And this is my last slide. So you will all agree with me, Ewing sarcoma is a rare disease, and so far all the projects has been made profited a lot by collaboration. So our effort is a multi-center effort. So far we have managed to collect material from over 120 patients from Vienna, from Germany. Uta and Marcus Metzler has contributed mainly on this. From France, as I already mentioned, from Didier and Olivier, and also from University of Oslo, where we have established a collaboration with Kitty Boyer. And all this work has been done within the framework of the Euro-Ewing Consortium. So thanks a lot. As I said, we are just started this project, and I know a lot of people are working in liquid biopsies, so it might be useful to talk to each other and coordinate our efforts. So unfortunately, we are not having a coffee break later on, but please talk to me or Uta and Marcus, and we'll be happy to work together. Thanks a lot.
Thank you so much, Eleni and Marcus, for beautiful presentations, and we really look forward to seeing how that translates into patient benefit, because actually this is something that we've all been looking for a very long time. I just want to say we're rather behind the schedule. We're about 15 minutes behind. So please can the next speakers try and keep to time, although it's so wonderful to be able to hear you speak, so we don't really want to stop you either. It gives me great pleasure to now introduce Paul Sorensen, who really doesn't need any introduction. He's a distinguished scientist and professor of pathology at the DC Cancer Agency in British Columbia, and has contributed so much to Ewing sarcoma. So thank you, Paul, for talking to us today about some metastatic features of Ewing sarcoma.
Thank you very much, Sandra and others. It's a delight to join this meeting, even at this ungodly hour for me. But I just want to go through some of our studies in Ewing sarcoma metastasis. Uta asked me to present our perspectives on this process. And I don't need to tell people on this call that the single most powerful predictor in Ewing sarcoma, of course, is the presence of metastatic disease. However, other than the fusions that we've heard about and a few other genetic alterations such as STAG2 loss, these tumors have a very low mutational burden, which has really made the identification of genomic drivers of metastasis very difficult. And I thought I would just give you a quick primer on the metastatic process and how this occurs in different compartments to sort of set up what I'm going to tell you in a little bit. So the first step really is migration and invasion of tumor cells into the local microenvironment, and then intravasation into the circulation. Then, of course, cells have to survive after detachment from the extracellular matrix, so-called suppression of monoicus, and then circulating tumor cells must transit to distant sites. And there, they must extravasate, migrate, and then adapt to new microenvironments, and then start proliferation all over again. So over the years, there have been a huge number of excellent studies to try to find the drivers of metastasis in Ewing sarcoma by many of the speakers that are part of this meeting. And many of these, if I go through all of this, have really focused on some of the early steps of metastasis, migration and invasion, or as we've heard about with STAG2 loss from these recent papers, reprogramming of fusion-driven transcriptional programs. And we got into this some years ago. First of all, we showed that the ERB4 tyrosine kinase activates a major migration factor, RAC1, to turn on invasion and metastasis, and also to block anoicus. We then showed that an RNA binding protein called YB1 translationally activates HIF1 alpha to allow Ewing sarcoma cells to adapt to hypoxia, contributing to metastasis. Then we showed around the same time that YB1 also activates formation of these granules that Olivier talked a little bit about, stress granules that can block oxidative stress and other stresses and drive metastasis. And then finally, more recently, we showed that YB1 can directly activate expression of the NRF2 antioxidant pathway to block oxidative stress and contribute to metastasis, and that class 1 HDAC inhibitors can actually block most of these processes. So this is great, but it kind of led us to ask the question, with all these different mechanisms, what's going on here? Is there some kind of common converging pathway that can explain all of these different findings and the findings of others? And so we took sort of a more holistic perspective on metastasis, and we remembered data that have been around for decades that metastasis is actually really inefficient. Only a very small minority of cells that start out to metastasize actually survive the whole cascade. And most of us that think about these sorts of things realize that there are a whole bunch of different stresses that occur along the way. And so we wondered, is there some sort of common converging stress or set of stresses that limit metastasis? And I'm going to give you a little bit of a spoiler alert here and tell you that we were really impressed and influenced by this paper from the Sean Morrison group that directly showed that oxidative stress inhibits metastasis in melanoma. And this has led us to really focus on oxidative stress as perhaps one of these defining mechanisms that can inhibit metastasis. And I just want to remind you that there are lots of ways that tumor cells are exposed to increased oxidative stress and reactive oxygen species. And the only way they can survive is by activating some of these scavenger antioxidant pathways. We've already heard about NRF2, but there are metabolites and dietary factors. And I just want to tell you a little bit about a recent story that really tells us that we're, I think, on the right track. This is work by Haifeng Zhang and Alberto Dele in the lab to this end. So what Haifeng asked is what we thought was a fairly simple question. He asked, what is it about pre-metastatic or successfully metastatic cells that allows them to turn off anoikis or detachment-induced cell death compared to normal cells that actually die when they detach, normal mesenchymal or epithelial cells? And he actually used a fairly simple method. He switched cells from 2D monolayers to 3D spheroids, because we know from the literature that this induces a very high level of ROS on the cells, including Ewing sarcoma. So this was the kind of setup. He transformed NIH3T3 cells with either mutant KRAS or ETV6 NTRK3, and then grew them in non-adherent cultures. And as you can see, not surprisingly, the transformed cells survived, but the non-transformed cells died. So he asked, what's going on right at this critical bifurcation point? He used proteomics to look for drivers of this anoikis suppression. And he came up with this really, actually a set of 31 proteins, but this one really caught our eye because it's a surface protein. And I don't have time to tell you today, but we're very interested in targeting this immunotherapeutically. But as you can see here in 3D cultures, particularly in ETV6 and NTRK3 fusions, this protein is very highly upregulated. So then, because we're a pediatric oncology lab, we asked which pediatric cancers have high expression of this IL-1RAP protein. And sure enough, Ewing sarcoma was right at the top of the list. So of course, we had to confirm that. We did immunohistochemistry, showed that there was surface expression. And I'll just tell you very briefly that we now know that IL-1RAP is a bona fide EWS-ETS fusion target. If we inactivate EWS-FLI1, IL-1RAP goes away, and this is a transcriptional response. And this led us to this recent manuscript from our group led by Haifeng Zhang and Chris Hughes and Alberto Dele identifying IL-1RAP as a surface targeting Ewing sarcoma. But I think what's really interesting about this is what this protein actually does in Ewing sarcoma and what it's telling us about Ewing sarcoma metastatic biology. So it's actually called IL-1 receptor accessory protein because in myeloid cells, it binds to the IL-1 receptor, but we could never show that it binds to IL-1 receptor. And so we did our own interactomics and found that among many interesting proteins, this protein CD98 is an interactor. And this is interesting because it binds to XCT on the plasma membrane. And this is part of the system X cysteine transporter, which brings cysteine in and pumps out glutamate. Cysteine is then converted to cysteine and eventually into glutathione, this major cellular antioxidant. And then immunofluorescence shows this nice co-localization with CD98 and XCT. And then we did another experiment in parallel. We asked which biological pathways might also be regulated by IL-1RAP. So we knocked it down using a couple of different shRNAs and looked for proteins that dropped out. And another protein, or one protein, really stuck out for us, something called CTH, or cystathionine gamma lyase. And I'll tell you what that is in a minute. But basically, inactivating IL-1RAP almost completely knocks out CTH protein, and this is what looks to be a transcriptional process. And so what does this all mean? So here's the XCT system that when there's lots of cysteine outside the cell, it gets pumped in, replenishes cysteine, which gets converted to glutathione to block ROS. However, if cysteine levels are low outside the cell, then the cell can actually make its own cysteine via something called the transsulfuration pathway for de novo synthesis. And this involves the CTH protein that I just mentioned. And so what we think is going on is the following. Fusions drive expression of IL-1RAP, which binds to this complex to ramp it up. It turns out that Ewing sarcomas have fairly low levels of XCT, so they're a little bit limited in bringing in cysteine. So if cysteine is actually low, then miraculously IL-1RAP switches to this other mechanism to drive de novo synthesis of cysteine. And so what we think is going on is that IL-1RAP is critical for maintaining cysteine and GSH pools in cells and acts as a sort of a rheostat between low and high cysteine conditions. And this really tells us that Ewing sarcoma cells are highly dependent on cysteine and GSH for maintaining redox balance. So just to show you that this is also going on in vivo, if we put these cells under the renal capsule of mice, immunocompromised mice, the Ewing cells are extremely aggressive, grow into the red normal kidney, but this is completely blocked if we inactivate IL-1RAP, and this correlates very well with loss of metastatic capacity. And these tumors, the primary tumors, have much higher oxidative stress burden and lipid oxidation, which we'll come back to. Similarly with CTH, if we inactivate this transsulfuration pathway, this also blocks local invasion and metastasis, which we can completely rescue by putting CTH back in. So what we think is going on is that in the primary tumor milieu and in metastatic cells, there's this interplay between enough cysteine being available to produce glutathione or the need for cells to make their own cysteine and glutathione. So there's one more piece to the puzzle that we find very interesting. There's this type of cell death called ferroptosis, in which if there's lots of mitochondrially derived ROS around, in the presence of iron, these go into something called the Fenton reaction to generate really reactive oxygen-free radicals that can oxidize lipids and break down cell membrane permeability and integrity. And this causes this entity called ferroptosis. This is counteracted by this XCT system that I just mentioned, because glutathione is a cofactor for something called GPX4, which can prevent ferroptosis. And so the way people study this is either blocking XCT with Erastin or blocking ferroptosis with iron chelators such as ferrostatin. And so what we noticed is kind of interesting. The Ewing sarcoma fusion protein is actually looks like it's preventing ferroptosis, because if you inactivate it with this TET-on inducible knockdown system and then treat the cells with Erastin to block XCT, this drives extremely high levels of ferroptosis, and these cells are highly vulnerable to death, particularly in 3D anchorage-independent cultures, which you can completely block with this iron chelator. So we really think that Ewing sarcoma cells are susceptible to ferroptosis, and that the fusion protein goes all out to prevent this process. So does this have anything to do with IL-1RAP? Yes, we think it does, because if we inactivate IL-1RAP, then they become exquisitely sensitive, just like Ewing sarcoma knockdown cells, to ferroptosis and lipid oxidation. And it's exactly the same thing with CTH knockdown. So we think that this kind of interplay between cysteine uptake and de novo synthesis is really important in Ewing sarcoma cells. As you can see, we can completely block this with ferroptosis. So what we think is happening in this context is that Ewing sarcoma fusions can drive IL-1RAP and perhaps other oxidative stress modulators, and through XCT induction or activation and CTH induction, make sure that there's enough cysteine and GSH around to block ferroptosis. And so to summarize everything, I hope I've convinced you that ROS is something that tumor cells face, and high levels of oxidative stress actually block metastasis, and that Ewing sarcoma cells and probably other cells have come up with all sorts of ways to mitigate this ROS induction, and that this confers survival under the many stresses of the metastatic cascade to actually facilitate metastasis. And so one of the big questions is there a dominant mechanism that Ewing sarcoma cells do this, or do these all contribute to metastatic fitness under different conditions? And so we're excited about targeting some of these pathways. I didn't have time to tell you that we're working hard on antibodies and antibody drug conjugates and CAR T cells to IL-1 MAPs to try to mitigate this process. So that's where we're going with this research. So I just want to stop here and thank the people that helped, particularly in green, Haifeng, Alberto, Chris, and others. Some of our friends in Vancouver, the Stand Up to Cancer group, of course, Didier and Olivier, who helped us with some of this work, and Stefan Burdek. So I'm going to stop there and thank you for your attention.
Thank you so much, Paul, for what an excellent presentation and another insight into what's modulating Ewing sarcoma metastases. In the interest of time, I'm going to move on again, taking a different tack. Uta's said in the chat, who needs breaks when you can have such fantastic scientists speaking to us? And I think there's definitely something for that. We want to allow you the space to give us your pearls of wisdom. So Katja is our next speaker, who also doesn't need any introduction. She's based at the Rizzoli Institute and has contributed in so many ways over the last few years. She's going to talk to us from a completely different perspective now, looking at exomes and microRNAs. So thank you so much, Katja, and over to you.
Okay, thank you. Thank you to everybody. I just, okay. Okay. So I hope you can hear me and see the slides. No? So thank you very much for inviting me to this interesting meeting. I am more than honored to be here with such a great scientist. And I'd like to provide some evidence of what we did to this fantastic and very active scientific community regarding exosome and their potential in terms of diagnosis and therapeutics. So to start with, you have already heard from all the other speakers that we have, sorry, you see the slide or just, okay. Okay. So you have already heard from all the other speakers about the importance of the chimera, EWS, ETS transcription factors, and you have very well, and since so many great labs are working on this chimera, we decided several years ago to focus on two different targets. And actually our interest was mainly on CD99 and on Igf-1 which is an RNA binding protein. So CD99, you know that there is whenever there is a Ewing sarcoma, there is also CD99. So it's Ewing sarcoma, it's very highly expressed in the tumor, and it is very nicely expressed at the cell surface of Ewing sarcoma cells. Igf-1 this is an RNA binding protein that is mainly located in the cytoplasm, and it can link hundreds of different RNAs, and actually it is a very powerful molecule that can regulate at a different level the fate of messenger RNA and microRNA of many targets. So to start with CD99, I couldn't miss this opportunity to speak a little bit of this molecule. Most of you know that this is my favorite molecule, but actually it is really a neglected molecule. And this is even inside the Ewing sarcoma community, that this molecule is often forgotten, despite the fact that we have quite a lot of evidence indicating that this is really important, either as oncosuppressor or as oncogene. And there are quite a lot of data indicating that this molecule can regulate crucial biological processes inside the cells, but also it can regulate the relationship between tumor cells with the tumor microenvironment. And why it is neglected? Probably because CD99 is really a unique molecule that essentially is unlike to any other molecule that we have known. And even if we studied the sequence and the structure of this molecule, it is really difficult to, this really don't tell us how this molecule is acting inside the cell. Despite that, we have demonstrated several years ago, that 10 years ago actually, that whenever we silenced CD99 in a Ewing sarcoma cell, even if this Ewing sarcoma cell have a perfectly functional EWS ETS chimera, we were able to shut down quite a lot of processes. We were able to limit and inhibit the tumor aggressiveness. So we were able to abrogate the spheroid formation. We were able to reduce the tumor growth also in vivo, and we were able to limit or to abrogate the formation of metastases. And on the other hand, we were able to induce cell differentiation, and in particular the expression of neural differentiative marker, even quite late differentiative marker. So this was done by modulating the molecules at a genetic level inside the cell. And these processes were obtained where we were able to identify at least some mechanisms that are related to this, and mainly these are related to modulation of MAP kinase, inhibition of Notch and NFKB signaling, in partly due to the overexpression of miR34A. So these are part of the mechanisms that are responsible of this phenotype. And as I said, these were obtained by genetic alteration of CD99 in at cellular level. But then we discovered that CD99 is released in cell supernatants, and it is released inside exosomes. And this means that in the tumor, we have Ewing sarcoma in a dynamic way. Ewing sarcoma cells can exchange exosomes that usually have CD99 on their surface. So we wanted to check what is the difference between between the communication that Ewing sarcoma cells have if they exchange exosome with or without CD99. And what we discovered was that if we expose Ewing sarcoma cells with exosomes that were deprived of CD99, we can recapitulate exactly the same phenotype that we have observed by shutting down the expression at the CD99 in Ewing sarcoma cells. So cells that receive Ewing exosome, CD99 negative exosomes, were inhibited for their migration capability, and they can differentiate towards and show quite a nice neural differentiation. So, and this is much more interesting, of course, in terms of a therapeutic perspective, because it's much easier to expose cells to exosomes than to modulate the expression of the molecule at the genetic level. So what are exosomes? Exosomes are very tiny vesicles, microvesicles. They were discovered many years ago, but for a long time, their function, we know very little about their function. It was only in the last, let's say, 10 years that we discovered that inside exosomes, we can have very important molecules. We start with messenger RNA, microRNA, but now it is well known that we can have also DNA and a lot of proteins. Many of them are very important in the signaling, in the signaling transduction. So there is no doubt that exosomes are important means of communication. There are some papers coming out recently describing the possible role of these microvesicles as biomarkers in the diagnosis and also in the modulation of phenotype. Mainly it was some paper that described a modulation in stemness or in migration. So two of these papers came from our lab, and we just show that when we expose Ewing sarcoma cells to exosomes with or without CD99, we can induce a massive modulation in terms of gene expression in the recipient cells. And at least among these signatures, we were able to identify modulation in transcriptional factors that regulate cell differentiation and cell proliferation, such as AP1 and the phosphotranscription factor. So you see that when cells, Ewing sarcoma cells, receive exosomes that were deprived of CD99, the activity or the expression of this transcriptional factor is decreased, and so it is the expression of some of the targets of AP1, like cyclin D1 or metalloproteinases. And this is probably or partly due to the fact that the exosomes that have or do not have CD99 on their surface and derive from cells that have or do not have CD99 have really a different cargo in terms of microRNA. So we were able to identify a signature that was differently differentiated these two types of exosomes, and in the exosomes that comes from cells deprived of CD99, so less aggressive cells, we have an overexpression, an increased expression of some microRNA, mainly oncosuppressor microRNA, which includes miR34A that we have already described, that was an oncosuppressor also in Ewing sarcoma, and it can increase the chemosensitivity of cells, and it can decrease the migration and the malignancy of cells. But we can also add to the list these two microRNA, miR199 and miR214. These two microRNA can act either as oncogene or as oncosuppressor, depending on the cellular context. In the Ewing sarcoma cells, we can say that these two microRNA can inhibit proliferation, can inhibit migration, and can inhibit the activity of AP1 FOS or the activity of this co-transcriptional factor AMGH1. And more important, the expression of these two microRNA were found to be lower in the metastases compared to primary tumors. So to summarize this part of the story, we can say that whenever we have a Ewing, whenever we deprive Ewing sarcoma cells of CD99, these cells started to release exosomes that are increased in some microRNAs, at least 3 microRNAs, and the nearby recipient cells can receive these microRNA, and these lead to a decrease in notch NKB signaling, ion C-phosphate 1, and also HMG1 signaling, and this results in lower growth, lower migration, increased differentiation. So this is one part of the story. The other part is related to this Igf-2 by BP3. This is an RNA binding protein. As I said to you before, a very powerful molecule. It is very interesting because it is not expressed in normal cells. It is de novo expressed in cancer. And so it has a great potentiality in terms as biomarkers, but also as in terms of therapy. We have demonstrated that at cellular level, whenever this molecule is highly expressed, the patients goes worse. So the survival was bad, and the expression of this molecule actually was higher, also in metastasis compared to primary tumor, in particular compared to to patients with localized Ewing sarcoma that go well. So this seems to be quite an important diagnostic biomarker. It is also secreted, so this was observed in other tumors, and we were able to confirm this also in Ewing sarcoma cells. So again, whenever the cells has high expression of Igf-2BP3, these cells release exosomes with high expression of this RNA binding protein. In contrast, when cells have a low expression of Igf-2BP3, they release exosomes with low expression of this RNA binding protein. And again, exposing recipient cells to exosomes that have high or low expression of this RNA binding protein recapitulates what we have observed in the cells by modulating the expression of Igf-2BP3. In particular, when Ewing sarcoma cells were exposed to exosomes that have a high expression of Igf-2BP3, we have a stronger migration, and we have a stronger activation of some of the recognized targets of this RNA binding protein, so Igf-1 receptor and 6CR4. And of course, we also have an increase in the activation of all the downstream signaling pathway. So I hope that I have been able to show you that even if these microvesicles has been disregarded for decades, it is now well understood that they are key elements in the communication between cells. They can have quite a variegated type of cargo that includes messenger RNA, microRNA, but also a lot of proteins. And for that, the exosome can be good or bad. It really depends on the context and on the cargo. But it's certain that, in my opinion, exosome cannot be discarded anymore, because they can be viewed as a powerful source for novel biomarkers, and certainly we need to study them more in detail and to explore more in detail if they can be used also for the treatment, since the immunogenicity and the toxicity of exosome was demonstrated to be quite poor. And also we know that these microvesicles can overcome physiological barrier, can spread very well in tissue. And it was very well demonstrated that this exosome can be loaded by drugs and protein and microRNA. And when this is done, the efficacy of the drug is really enhanced. So since exosomes are usually schematized as bold, is with these small balls that I like to thank you for listening to me. Thank you.
Thank you very much, Katia, for an insight into another important piece of the puzzle in terms of modulation of Ewing sarcoma, and also bringing up potential for additional biomarkers, importantly. So thank you for that. We're going to now move to the final speaker. Again, we're over time, but wonderful to be able to hear these insights. I'd like to welcome Sue Birchall, who's Professor of Cancer Biology at Leeds University, who's also going to give us some insights into some cells responsible for progression and potential biomarkers. So thank you very much, Sue.
Thanks very much. Can you hear me? Hello, can you hear me?
We can hear you loud and clear, thank you.
Can you see my slides?
We certainly can.
Okay, are they in presentation mode?
Yeah, they're in your slide sorter.
Oh gosh.
No, they are not.
Oh gosh.
Go to the top.
Go on.
To the top next to the save button.
For some reason I can't get there, which is a bit frustrating. We tested this before and it was fine.
Along the bottom. Yeah, try that again. It doesn't look like it's working, does it?
Hang on.
Maybe there's a delay.
There you go. There you go. Is that okay?
Yeah, perfect. Well
okay,
done.
Thanks very much for your patience. Sorry about that. So we've heard a lot today about clinical heterogeneity of Ewing sarcoma and the challenges that this brings, and the strategies that are being taken sort of across the globe to improve outcomes for patients. In Leeds, we've been studying the biology that underlies the heterogeneity of response or non-response in patients. So trying to use this biology, understand the biology of the cells that are actually resistant to current treatments and that may have the capacity to self-renew, and so therefore may be responsible for progression and subsequently relapse. And our hypothesis is that these so-called Ewing sarcoma stem-like cells, if we can find markers to detect these cells and candidate targets to treat the cells, if we combine treatment against these cells with standard chemotherapy in a combination treatment strategy, that we might actually be able to develop more effective kind of treatments. So there's been a number of different strategies used to identify these drug-resistant self-renewing cells. And most frequently, people have used cell surface expression of proteins, particularly CD133, to identify these cells in a number of different cancer types, including Ewing sarcoma. Unfortunately, although CD133 positive Ewing sarcoma cells certainly do have self-renewing capacity and are capable of migration and metastasis in mouse models, not all the sort of self-renewing drug-resistant cells that we believe are capable of repopulating tumours are actually expressing CD133. So in the work that I'm going to tell you about very briefly this afternoon, what we've actually used is a functional approach to isolate these self-renewing chemo-resistant cells. So we've actually worked with a panel of patient-derived Ewing sarcoma cultures, which have been established and characterized by Liz Roundhill, who's a postdoc in my lab, who I'm sure many of you will know of. And from these patient-derived cultures, Liz has actually used 2 different characteristics. So initially, seeding multiple Ewing sarcoma cells into 96 well plates, so looking for single cells and the capability of these single cells to produce clones in a sort of 96 well plate. So for each experiment, she'd seed 960 cells from any one patient-derived culture. And then after 3 weeks, look to see which cells had actually managed to grow and produce a clone. And then where possible, she selected those cells and propagated them. Confirmed that these cells with this self-renewing, single cell self-renewing characteristics are actually resistant to common chemotherapies that we currently use in the clinic. So you can see just there's some plots where there's the response to doxorubicin and etoposide. The blue dots, the blue curves are actually the response of cell lines to these particular chemotherapies. And you can see to the right-hand side of that, the grey and the black lines are actually some of the patient-derived Ewing sarcoma cells, ESCSCs, that are clearly much more resistant to standard chemotherapies than the cell lines that we normally would work with. One of the striking findings from Liz's study was that we were always able to find at least one cell that was capable of single cell self-renewal from all the tumours that we investigated. So I think this is some really compelling evidence that Ewing sarcoma is a cancer stem cell-like driven cancer, and this kind of supported or gave us confidence in our hypothesis to try and find treatments to eradicate these cells. So having identified self-renewing drug-resistant ESCSCs, Liz went on to use total RNA sequencing to characterize these cells. And not surprisingly, when we look at the transcriptome of patient-derived cell cultures from established cell lines, then this is very different, and the patient-derived cell cultures cluster independently of the cell lines. This work has all been published in this paper in Cellular Oncology earlier this year. And we're now using these patient-derived cultures in a number of different strategies to identify novel therapeutic targets, biomarkers of risk, to actually run drug screens, and obviously to develop multicellular models. This afternoon, I'm just going to talk briefly about some of the biomarker work and that's been published, and some of the preliminary work from the drug screens that Liz is now undertaking. So when we looked at the transcriptome of the patient-derived ES cells and compared this to the patient-derived ESCSCs, we found a huge number of differentially expressed genes, so 561 differentially expressed genes. But when we looked to prioritize the genes that were most significantly differentially expressed, so looking for things that were either changed by at least two-fold increase or two-fold decrease, then we were able to reduce that number to something that was a bit more manageable to study. The details for all the sequencing are available online. There's the link there if anybody's interested in looking at the data. But what Liz did was she identified a panel of markers that were increased more than twofold significantly in ESCSCs compared to the Ewing sarcoma parental cultures, and validated or used PCR and immunohistochemistry to validate the expression of these markers at the RNA and the protein level. And as Tomas very kindly highlighted earlier on in the afternoon, one of the striking markers that Liz found was neurexin-1, which high levels of which identify patients who seem to do particularly badly. This is high levels of the neurexin-1 protein expression detected by immunohistochemistry. You can see an example of a low expressing and a high expressing tumor on the bottom there. I think this was actually the plots that Thomas showed in his presentation. These are just looking at the expression of neurexin-1 in patients with localized disease, and you can see this large separation of patients who are doing badly, seem to have an outcome similar to those of patients with metastatic disease compared to those with a better outcome with the lower levels of neurexin-1. These results are in a very small group of patients. You can see there underneath the Kaplan-Meier plots, the actual number of tumors that have been examined. So a priority for us now is to investigate these markers in a larger cohort, and in collaboration with Enrique, Didier, Katia and Laura Curtin, who's a statistician from the CTU in Birmingham, we're actually evaluating the independent prognostic value of neurexin-1 and other biomarkers, including IGF-2 BP3 that Katia's just been talking about, and expression of STAG2, which Didier's published on. The idea behind using neurexin-1 then, or indeed whichever panel of biomarkers allows us to separate children, young people at greatest risk from their disease, is that at diagnosis, we might be able to provide tools that clinicians can use to improve the stratification of young people for treatment. So particularly, I think this ability to identify at diagnosis that 30% of young people who have seemingly low risk disease, but actually have the similar outcomes to patients that are actually presenting with other metastatic disease. And we'd hypothesize that with patients with localized disease, but high expression of neurexin-1 might then benefit from randomization to treatment for metastatic high-risk patients. This study with Enrique, Didier, and Katia and Laura has been a collaboration through the EEC, and there's the link on the bottom there to find out more about the EEC network, if anybody wishes to look into that and join the network. So apart from identifying the patients who are at increased risk and providing potentially clinically informative new biomarkers, neurexin-1 and the pathway that it regulates may well be a candidate therapeutic target, although this is less well evidenced, certainly in Ewing sarcoma. Neurexin-1 is actually expressed in the presynaptic membrane of cells and is really important in cell-cell communication. And it has a functional role in development of neural disorders that are characterized by this sort of synaptic dysfunction. So while it's not clear whether neurexin-1 is a good candidate therapy at the moment for Ewing's, there certainly are tools out there and potential treatments that target this pathway that could be exploited if this proves to be of interest. So just very briefly, in a couple of slides, one of the other things that we're doing with the primary cultures is actually carrying out drug repurposing screens. And here we're using high throughput screens using imaging systems to screen the primary patient-derived cells, the primary cultures and the ESCSCs for drugs that have already been in patients and that we already have quite a lot of information about. So again, this work is being carried out by Liz, and Liz has actually screened a panel of ESCSCs and identified 75 out of 1,290 drugs, which actually are significantly more effective at decreasing Ewing sarcoma cancer stem cell number than doxorubicin. And we've kind of used this as a marker to find candidate agents that we might then be able to combine with chemotherapies. So at the moment, Liz is working on looking at the synergy, whether there's synergy or additive or even antagonistic effects between some of the agents that she's identified as killing ESCSCs when they're combined with each other, and also when they're combined with the sort of common chemotherapies that are used in the clinic to treat patients. Just give you a flavour of the sorts of agents that we're finding particularly interesting. When we've looked across the board, actually a large number of agents that are inhibitors to the aurora kinases are particularly effective. You can see on the bottom left-hand corner, when we combine CD532, which is an aurora kinase inhibitor, with ifosfamide, then we actually see a synergistic activity decreasing viable cell number. And this work on the aurora kinases and inhibitors of aurora kinases is being taken forward by Molly McNee, who's a PhD student in the labs. And this table just summarizes the effects on 4 different Ewing sarcoma cultures, patient-derived cultures, and where you see a green box, that effectively means that there is synergy between the inhibitors. In this particular table, we're looking at 2 different aurora kinase inhibitors and the different chemotherapies that you can see there. So I think this is not published, this data, but it gives us some confidence that targeting or combining drugs that target ESCSCs with chemotherapy that's already used in the clinic may well be a promising strategy. So I think this strategy works from the scientific perspective. We've been able to identify novel biomarkers that are separating patients into risk groups that previously we haven't been able to identify. And certainly we've identified some candidate therapeutic targets, and using the cells in a drug screen, we can identify existing drugs that may well be worthy of evaluation in early phase clinical trials. Whether or not these results and this strategy improves outcome for patients, either by the introduction of more effective therapies or the introduction of kinder therapies. Obviously, we need to test these things in the clinic, but we hope to do that once we've identified most effective therapeutic combinations and biomarkers that would allow us to select the patients. So yeah, that's certainly a strategy that has worked scientifically and in preclinical models is promising. The next steps over the next few years are to find the best combinations to take into trials to see if this really does improve outcomes for patients. So just like to thank everybody who's contributed to the study, in particular the patients and families that have given us samples for the research, because without those primary cultures, we wouldn't have been able to do this at all. And of course, the many funding bodies that have supported our research. Molly at the moment is funded by Sarcoma UK, they're funding a PhD, and Liz is funded by the Little Princess Trust. although we have been funded in this work previously by BCRT and the Ewing Sarcoma Research Trust and CCLG.
Thank you so much, Sue. Again, apologies.
Can I just show this one last slide, Sandra? It's just to ask people to save the date, because we are hoping to have an in-person meeting next year, COVID and travel restrictions allowing. So
thank you, Sue. That's great. Can we share that with the group later, if you don't mind? I'm really sorry. We are very behind time. Really wanted to give you all an opportunity to share your insights with us. Thank you, Sue, also. And just highlighting the number of biomarkers that are coming through. And it's something that we're going to discuss at the EEC meeting in January, about how we may be able to cross-validate using the clinical trials across Europe and perhaps beyond. Again, just want to thank the speakers and for the organisers. Apologies for the delay. We're now going to take a 10-minute break. It's 17:08 European time, so I suppose that would be 17:18, and clocks adjusted according to where you are. Thank you once again to everybody.